Hello, everyone. My name is Deng Jingyu. I am a Master by Research student in NTU. My major is Material Science and Engineering, and my supervisor is Prof. Cho Nam Jun. Today, I'm going to share my life and research in Singapore. First, let me introduce myself. I come from China, and my hometown is in Yichang City in Hubei Province, which has a very famous building named Three George Dam. Hope you have ever heard of it, and it is mainly used for electronic generation and to support the whole country. And I studied my undergraduate in Beijing Institute of Technology from 2015 to 2019 in Beijing, the capital city in China. And I started my Master by Research student from 2019 in Singapore in Nanyang Technological University until next year. First, I introduce my undergraduate student life. My major is electronic packaging technology, which belongs to material science and engineering in BIT. Beijing is a very famous city, and it has a lot of places of interest. The most famous one is one of the most famous one is the Forbidden City, which is also an imperial palace of imperial in Dai and in Qing and Ming dynasties. And also another very famous building is the Great Wall. And my life in Beijing is very colorful, except for the courses. We also did a lot of experiment and research assistant in the lab, and I also joined a lot of programs. One of them are as a volunteer to protect the environment in the grassland. And also I visit the Tian technology company to learn the process to fabricate the chips. Here are some photos for the ceremony of my undergraduate. And then start from last year, I came to Singapore, study as a Master by Research student here. 70% of my life in Singapore stayed in the lab to do some researches, and the other part I spent a very great time with my friends and lab mates. We cooked together and we visit a lot of places in Singapore, and such as the St. John's Lands in Singapore and such as the aquarium, and also through communicate with people from different countries such as Italy, Korea, and Myanmar. We learn different culture from different countries. And also I joined AKC last year in November. We spent a very great time, around 4 days there in the Marina Basin. And we listened a lot of seminars from people in different areas and from different countries. And also we had a great dinner there. We communicated a lot there. I really learned a lot from AKC last year. And for my research, I started to do some independent research project in Singapore as a master student. The first one is about the biodegradation of communal paper like pollen sheets. The pollen has a very special structure, which has 2 layers, the axine layer and intern layer. The axine layer is mainly composed of sporopollen. The intern layer is mainly composed of cellulose, pectin, and so on. As shown in these figures, we can see outside the axine layer, there's a pollen cement, which is mainly composed of lipid, and the shell structure protected the genetic materials inside the pollen. pollen has a lot of advantages, such as the shell structure, the two layers are ultra tough, which can protect the genetic materials in harsh environment. And also pollen has very uniform size for different species, and the structure for each the species for the pollen from the same species, they have uniform size, and their structure are very similar. Thirdly, because of the large amounts of production of pollen each year, so the price of pollen is also very low. For this experiment, we did a pollen paper preparation and the degradation experiment. The pollen paper preparation is mainly composed of 4 processes: defatting, cooking, incubation, and drying. Defatting process is mainly to remove the lipid and other contaminants. The lipid components mainly outside the exterior. Cooking process is mainly to remove cytoplastic components. And the incubation process is mainly used for further transform hard pollen particles into soft microgel. Drying process is mainly to dry the pollen gel to fabricate the paper-like pollen sheets. And for the degradation experiments, I treated the pollen sheet. I cut the pollen sheet into small pieces and treated them with DI water and PBS solution. The PBS solution is a kind of simulation of in vivo environment of the cell, and also I will try to characterize the pollen sheet every week in the first month, and every month start from the second month. Each time I will take out the pollen sheet from the solution and measure the weight and characterize FTIR to check the chemical component difference, to using FESEM to check the surface difference, and using surface morphology, and using the DMA to check the mechanical properties. For this experiment, we get a main conclusion here. Biodegradation process was observed for Camellia pollen sheets treated with PBS solution after 5 months through FTIR and SEM. Degradation mainly happened in lipid and carbohydrate component of pollen through FTIR analysis, but the mechanical properties stay stable for a long time. Pollen sheets-based electronic devices and actuators have a long-term degradation, which provides stability for implanting bioapplications. So because of these great properties of pollen, we want to replace the plastic using the pollen particles or pollen gel or pollen sheet. Because plastic, there's have a connection with another project, replacement of plastic and plastic leakage. Plastic particles really, people really exposed to plastic particles in our life, such as through different ways, such as inhalation, ingestion, skin exposure, and implantation, and which will cause the plastic particle bioaccumulation in human body. And this will have a very bad impact on human health, which cause a lot of disease, such as respiratory problem, cardiovascular disease, nose problem, cancers, and digestive problems, and so on. So in this research, I mainly treated the different kinds of food container productions, such as disposable water cup, coffee cup, styrofoam matchbox, plain wrap, which are very common in our life, but we use for food containers, food treatment. And we mainly study using those production to heat it in the DI water at around 95 Celsius degree to check the particles released from these productions. We can find, we observe a lot of particles, and we check the intensity distribution, the size of the particles, the amount of particles. Here about my future plan. There are several very famous words from Albert Einstein, one of the greatest scientists. You need to know the essence of scientific methods. Don't listen to what a scientist says to you, but watch carefully what he's doing. A man who never makes mistakes must never try anything new. Because I want to be a great scientist in the future, and I am on my way to learn how to do experiments, learn how to do the planning for the experiment, and how to how to fix the issues every time I met. Although sometimes I made mistakes, but I believe this is a kind of way to help me to gain more experience to become a great scientist. And I will always contain huge passion for the research. Thank you.